Dear students, Assalamu alaikum and welcome back to Plant Physiology course. In a series of videos, we have discussed diffusion and various aspects of diffusion. Now we are going to start our next section or chapter, and that is osmosis. So we are going to discuss osmosis, water potential, and various components of water potential and its relationship with the plant. And then we are going to move along and discuss the absorption of water by plants. So first of all, let's define osmosis. Osmosis is basically a diffusion. Osmosis is a special type of diffusion in which water molecules, that is the molecules of the solvent, they move across what a semi-permeable membrane, a differentially permeable membrane or selectively permeable membrane. From a region of their higher chemical potential, a concept with which we are quite familiar now. So, osmosis is a special type of diffusion in which water molecules move across a semi-permeable membrane from a region of their higher chemical potential to a region of lower chemical potential. So there must exist chemical potential gradient, and if there is no gradient, obviously the net movement of molecules of water across the membrane will be same. So we cannot say that osmosis is taking place as there will be an equilibrium. So when there is a gradient, so there is a net movement of molecules from one side of the membrane to another. So this was a definition. Let's go through it again. Osmosis is basically a diffusion, and it is a special type of diffusion because water molecules are involved. Now we can describe osmosis in terms of any solvent, but since in plants the only solvent that is absorbed and which moves throughout the plants, which is conducted throughout the plants, and in which various solutes are dissolved, that is water. So we are mostly concerned with water molecules. So water molecules, they move across a membrane, and now this membrane, there are certain constraints as far as this membrane is concerned, and that is it must be a semi-permeable or a differentially permeable membrane. So why they move, what's the reason behind them? It's the same reason which we saw during the discussion of diffusion. That is, they move from the region of higher chemical potential to a region of lower chemical potential. So when there is a membrane, so obviously there must be a chemical potential gradient across the membrane. So one side of the membrane may be at lower potential and other may be at a higher chemical potential. So the water molecules will travel, or they will move, or they will diffuse across the membrane from a region of a higher concentration to a region of lower concentration. Now this was the basic definition of osmosis. Now we can demonstrate osmosis by a special kind of apparatus which is known as osmometer. So we can demonstrate the process of diffusion with the help of an osmometer. Now you can construct an osmometer in the laboratory. It's very simple, that if we take a tube and tie some semi-permeable membrane, like a membrane taken from an egg, a chicken egg, so that membrane is a differentially permeable membrane. So how we can obtain that membrane, its discussion is beyond the scope. So you can tie it to the one end of the tube, and then you add a sugar solution, for example, or a salt solution inside the tube, and then you immerse this into a beaker of pure water. So this is an osmometer. So let's draw a diagram of an osmometer and see how osmosis takes place in that osmometer, and it can be very easily demonstrated in the lab. Let's construct an osmometer. In the lab, we mostly make use of a special type of funnel known as thistle funnel. But you can use any type of tube. The thistle funnel has a bell-shaped mouth and a long tube. It is somewhat like this. So this is a very ugly thistle funnel that I have drawn. Anyway, thistle funnel is somewhat like this. So we invert it, and what we do that we tie that membrane on the mouth of the thistle funnel, and then it is inverted upside down, and then it is immersed in the solution. But you can use any tube that is available that has sufficiently large bore so that we can easily add our solution. So here is the osmometer. It's a stand. We are going to just draw the bare bones, and here is the beaker of water containing distilled water, and then this is our tube which has been immersed in the solution, and we have tied that membrane, and this is the clamp which is holding the tube, and we have added a solution. So it contains, let's make it of a different color. So it contains a solution of sugar, sodium chloride, or any salt. Now this is a very simple osmometer, a glass tube or a thistle funnel turned upside down like this. And then you tie that piece of membrane at its mouth or on its mouth and close it, and then you add the solution and fill it with the solution. So now it's containing that sugar or salt solution, and then it is placed in the beaker of water. So this is beaker of water containing distilled water, and that thistle funnel has been placed in it. So whatever the case might be, this is a demonstration of an osmometer. So what you will observe, as we have seen definition of osmosis, that it is a movement of water molecules from a region of higher concentration to a region of lower concentration across a semi-permeable membrane. So now this is pure water or it is distilled water, and this is the solution of, say, sucrose. And you can add, for example, let's say it's a 5% solution of sucrose. So 5% solution means 5 grams of sucrose in enough water to make 100 ml of solution. So you have added 5% sugar solution. This is pure water. Now what happens that water will move from a region of higher concentration. So where is the region of higher concentration? This is pure water. So pure water means all water molecules are free. As far as this sucrose solution is concerned, some of the water molecules are attached or they are bound to these sucrose molecules, because when solvation, the process of solvation or solution forming takes place, some of the free molecules of the water, they get attached to the solute molecules, because the solvation process reduces the free energy of the solvent. So now in this particular solution, sugar solution, the number of free molecules are less as compared to the beaker of water. Here, all the molecules are free. They are totally free. All the molecules of water, they are free. So we have maximum free energy or maximum chemical potential here inside the beaker. So here we have maximum chemical, let's say Cp, chemical potential, maximum chemical potential. But in this solution, the amount of chemical potential has decreased. So we have lower chemical potential as compared to the beaker of water. So we have a higher maximum, or we can say this is higher chemical potential as compared to this tube. Now that we have seen that where is the region of higher concentration and where is the region of lower concentration. So I repeat again, in the beaker we have a higher chemical potential, higher concentration of water molecules, or greater free energy or higher free energy. But in the solution or in the tube, the amount of free water molecules is less. So we have a lower chemical potential and we have less free energy as compared to the water in the beaker. So now that we have seen where is the higher concentration and where is the lower concentration, so water will start diffusing across the membrane, so osmosis will take place. And how you will figure out that osmosis is taking place, because as water starts moving into the tube, the level of the solution inside the tube, it will start rising. So it depends upon how much solute is present. So according to that, the water will move inside and this level will start rising inside the tube. So this indicates that water is moving into the tube. So this is an osmometer and it simply demonstrates the process of diffusion of water molecules across a semi-permeable membrane from a region of higher concentration, which in this case is the beaker, region of higher concentration, to a region of lower concentration, which in this case is the tube. So water molecules will diffuse across the membrane. So this was a demonstration of osmosis and movement of molecules across the membrane. So more about osmosis and various concepts of osmosis we shall discuss in the coming videos. Thanks for watching and see you in the next video.